All right, guys, we'll be starting now. Before we go in, I would just like to let you guys know that today is going to be an introductory session into bioinformatics. And you will actually be learning this in your curriculum also for those who have taken biotech, BBT, that is BBT, guys, you'll be learning it in second year. And I think integrated MSI students, you'll be learning it in third year. So we just want to give you a very small introduction today into some basic bioinformatic tools. And in the upcoming sessions, we will be utilizing these tools systematically in order to solve some case studies. So the first two sessions are going to be to introduce you guys to these tools. All right. So before we move in, can I know how many of you guys are familiar with any bioinformatic tools or have any prior experience in this field? Anyone, guys? Hello, can you guys hear me? All right, so okay, give me a thumbs up if you guys have any experience in this field, or do you, if you know something about it already. All right, how many of you are new to this? Can you guys give a show of hands for all those who are new to it? All right, no worries, guys. So today's session will fully be focusing on the basic tools only. Okay. So please feel free to stop me in between if you're not able to understand anything, and I'll try to explain my best. Okay, to you guys. Now I'll be sharing the screen, so just let me know if you guys will be able to see it. Can you guys see my screen? All right, guys. Okay, so first we'll be moving to our first bioinformatic tool, and this is called NCBI. I'm sure many of you guys have an idea about what NCBI is, but for those of you who don't, we'll be going into it today. Okay. If you guys want to follow along with me, that's fine. Or if you just want to pay attention, also that works. Because it's only a very basic session, you wouldn't need anything hands-on to do along with me. So you guys can just note down whatever sort of tools I'm using, and later on go explore it. Okay. So NCBI is known as the National Center for Biotechnology Information, and this has a variety of information in it. This is maintained by NIH, which is the National Institute of Health, and this has the curated content of almost all the biotechnological molecules, the nucleotides, proteins, etc. So in this software, you'll be able to get a variety of information, and the most commonly used ones are the ones given on your right over here. Can you guys see my cursor move? Can you give me a thumbs up if you can see my cursor move? All right. Okay, so these are the main softwares which are actually part of this tool. And every one of these is going to be important in one of the other informatic applications in the later times. You guys will get to know it in the upcoming sessions. But today I'll just be introducing you guys to what these actually do. And I'm sure most of you guys have this habit of going through research papers, etc., and they will open up in PubMed. Okay. So in PubMed, you can give an input any keyword which you want about any journal you want to go through. Say for example, we want to go through any new recent articles that have been coming. You can go to the trending articles. So here you have a variety of articles which are given that have been published recently or that are currently trending. Okay. So for example, I want to uh, just one second. Yeah. Okay. So I want to research something about neurobiology. So I'm just entering the keywords over here, and you can go into this search button, and you will get a variety of articles. The thing is, PubMed is usually used to view research articles, but there's another thing called PubMed Central. You guys will see it over here. So this is called PubMed Central. The difference between these two is that in PubMed Central, you will be able to fully view the articles and you'll be able to download the entire article. You know how some of the articles are actually locked or we won't be able to see, so we would have to try going into any other tool like Sci-Hub or anything to view those articles, right? So in this, similarly over here, say for example, when I'm going to neurobiology, I can see that only some of the articles here will be written as PubMed Central article. So here you can see it is a free PMC article. All these PMC articles mean you have full access to these. So if you go inside, you will be able to visualize it in PubMed Central, and you will be able to view the entire article. But if it is not mentioned as PMC, say for example this one, here it is not mentioned as it is a PMC article, so you only have to go to whatever is available over here, and you may or may not get the entire article. So here, for example, see, it is showing you have to access through your organization, and you won't be getting access to this entire information. So that is the only difference between PubMed and PubMed Central. So PubMed will give you an idea about what all articles are available about that particular topic or field. And if you want to go further in depth into it, you would have to go into PubMed Central. So I hope you guys have understood what the difference between these two tools are. Bookshelf, we won't be using it much because it is much more of a medical related field where you can actually find a lot of books published over there. It is not necessarily medical, but usually those are the people who will use this particular tool, so it won't be of much use to us. Blast, actually, Blast you will be covered in the upcoming sessions, but I'll just give you an idea about what it is. So basically, this is an alignment tool. Once you go into this, you will have two things, which is a nucleotide blast and a protein blast. Alignment tool, basically what it does is it will compare your query sequence to an already existing sequence, which will be the reference sequence, and say how similar these two sequences are. Say for example, I am taking insulin genome sequence, okay, and I want to blast this against the reference genome and see what all genomes have similarity to my particular genome, which is insulin. So that is where your nucleotide blast will come in when you want to go compare the nucleotides. When you want to compare nucleotide similarity, if you want to compare protein similarity, that's when you look for protein blast. So I won't be going much into this because this will be covered in tomorrow or sorry, the next session or the upcoming ones. So I won't be going much into it, but just for you guys to know what blast is. So it is just an alignment tool to see how close your sequence is against a particular reference sequence. Okay. Next one. So these three web tools are actually to obtain the nucleotide sequence or the genome or a single nucleotide protein of a particular organism which you want to look for. Okay. Let's say I want to go into the nucleotide. Okay. Once you go in here, you can type out anything you want. So, I mean, since we took the example of insulin before, we can write insulin and you will automatically get it as Homo sapiens. And it is very important that you mention which organism you are looking for because there will be a variety of sequences available here, but you would have to mention which one you are looking for so that it will be more specific to your search. And in this column, it might be a bit difficult to search. So in the right side, you will have the top organisms and there select for Homo sapiens. Okay. And once you get that, you will have multiple options available here. Usually what we would need is the complete CDS. CDS means it's the coding sequence. Okay. So this will be the coding sequence for the insulin gene. And we would need a complete CDS. Complete CDS will include both the coding and the non-coding regions of the DNA, and inside that we will be able to find which one is actually responsible for producing the insulin protein. But it also will include your other regions such as the UTRs, the transcription regions, etc. So all of these will also be included. Just a second, guys. I think it's taking some time to load. Okay, anyways. So once it loads, I will show you guys what exactly this is. So until then, I will open another browser and show you guys the rest. Okay. Before I move on, actually, I would just like to let you guys know one thing. So when we want to take in the sequence, right? That is the complete sequence which is shown here. That is the CDS. So below it, you will be able to find the sequence displayed. Here you can see. Okay, we got it. So this is the entire sequence of the insulin gene. Okay. So you have so here you will have information on how many base pair sequence it is. So here it is 4969 base pair. It is the DNA. It is a linear DNA, and this is called as the accession number. So the first part is the accession number, and the point something is the version number. GenBank is actually the repository which will be able to submit these data to you. So it will verify all the data which is submitted, and it will be displayed over here. So GenBank is the repository which is showing all of this. Now you have so many other informations over here, such as uh, the source organism, and who are the authors who contributed towards this, etc. Now, what we would require is among this entire sequence, not all of it will be coding for the insulin protein. Only the CDS part will be coding for the insulin, and when we select that, the region will be highlighted. Just give me a second, guys. Yeah, I'm sorry. So yes, so when you click on the sequence, your coding sequence will be highlighted. So this will be the one which will code for the actual insulin gene. The rest of the sequences, they are there and they perform other functions like for transcription factors, for untranslated regions, alternative splicing. They have so many other rules, but this is the only one which will get transcribed to an mRNA. So now, if we want to get this entire FASTA sequence, you may ask, if this is only the coding sequence, then why would you require all of this, which is unnecessarily there? Usually, the CDS, the complete CDS, is utilized when we want to design primers. When we are performing PCR to amplify your fragments. In those cases, you would have to design a primer. A primer it has to be such that it covers the entire sequence, and that should be from the start till the finish of the entire genome. Only then you will be able to amplify the DNA. So, for all your polymerase chain reactions, when you want to design primers, you will go for the complete series. And as I was mentioning before, FASTA, when you go to the FASTA, it is a format in which it is stored. Okay, so FASTA format is one where you will be able to download this particular sequence. Okay, so yeah, it is taking some time to load. When it loads, I will show you guys that. Until then, we can move on to the next one. Same with the genome, same with SNP. So in all of these softwares, you'll be able to visualize the genome of a particular organism, the single nucleotide protein. So most of these, we won't be utilizing it much. Same with gene or protein. So all of these softwares, we won't be utilizing it much, because mostly, if at all we are using NCBI, it will be for PubMed Central, BLAST, and Nucleotide. In rare cases, when we do want to look into the entire genome of an organism, you go for a genome database. And then if you want to look at a particular gene, say only the insulin gene which is coding, you can go into the gene. And protein, that is for the insulin protein, not the gene. So when we go for the insulin protein, you can go into this protein database. So you have many such database, and these are only the popular ones. And there are so much more, and you can look at those over here. I won't be going into the details of all of these today, but as and when we are going to the other sessions, and when we will be requiring any of these, we will be introducing you to all of these softwares. So you just have to know. Yeah. So when I clicked on the FASTA format, this is what I got, right? So what FASTA actually means is, it is a format where you will get the entire sequence of the DNA, and it will start with this symbol. Okay, it will start with this symbol, and here you will have the name of this particular sequence. Okay, so here it is mentioned that this is the accession ID, this is the version, and this is the name of the sequence. When you want to design your own faster format, you just have to put this symbol. So yeah, this symbol you have to put, and you can give any name you want to your sequence, and then below that you will have your entire sequence. Okay, so this is essentially how NCBI works. Do you guys have any other doubts so far? Do you want me to clarify anything? If you don't have any doubts, please give me a thumbs up. All right, that's great, guys. So please feel free to stop me in between if you guys have any other doubts. Okay. All right. So now I hope you guys have understood what NCBI is. It's a curated website which will have a lot of information. It is not limited to any particular thing. So as I just mentioned, it is for biotechnological information. Now we'll be moving to something a bit more specific and bit more commonly used, and this is called UniPROT. Okay. So now UniPROT is a bioinformatic tool which will give you information about your protein sequence. Okay. So we'll be using another tool also, which is PDB. PDB is a protein database. Okay. Sorry, protein data bank. And in this, so the difference between UniPROT and PDB is that UniPROT gives you sequence information of a protein. That is what all the amino acids which make up your protein. Okay, but PDB will give you your three-dimensional structure of the protein. For example, how many alpha helices are there, beta sheets, what is the structure, the three-dimensional structure of your protein, what is its molecular weight, and what basis they have found it. So that will be PDB. So that will be the difference between UniPROT and PDB. Okay. So I'll explain about PDB a bit later. Now we'll go into UniPROT. Okay. So, as I mentioned before, UniPROT is for identifying the particular, for getting information of a particular protein sequence. Okay. So, as is mentioned here, we'll be taking an example of insulin itself. You may enter any protein you want. Today, I'll be taking the example of insulin only. Okay, so that we can have a more consistent format. That's why. Okay. So when you search for insulin, this is what will be shown. Okay. Uh, yeah, you will be getting it in a table format. You may view it in cards format. You can view it any way you want, but a table format makes it much more easier to visualize it. Okay. So now in this, uh, again you can classify into organism, whichever organism you want, but we'll be going for the Homo sapiens one. Okay. Here, this whatever ID given here is known as your UniProt accession number. Like how you add it in your NCBI also. Every database will have a particular accession number for each of its entry. Okay, so this will be your UniProt accession number. This is the name of the particular protein, and this is the gene name of the protein, and it is in which organism it is present, and the length of it, how many amino acids make up this protein. Okay, so since we want the insulin gene of insulin protein of humans, we'll be selecting this entry. Okay, so usually it will give you based on your top searches only. That is, most default searches will include Homo sapiens, but you can choose whichever organism you are looking for. Okay, so now when you go inside this, this will be the information which will be displayed at first. So just to give you a brief on what each of this means. So here this gives the protein name, which is insulin. The gene is called INS. It is represented as INS, which is the insulin's gene. It makes, I mean, 110 acids make up this protein, and here it shows that it is UniProt KB reviewed a SwissProt. So here in UniProt, there are two main things which you guys should know. One is SwissProt, and the other one would be called Tremble. Tremble, which is spelled as TREMBL. Okay, that is Tremble. The difference between these two is that SwissProt is a more manually curated version. So, for example, someone has entered the sequence for this insulin gene. There have been so many verifications by so many scientists. It has been manually checked and verified, and then it has said that yes, okay, this is your sequence. So that is what SwissProt does. So if something is verified by SwissProt, it means it has very high accuracy, and it is very close to, if not 100% also, it is at least 98, 99% correct sequence only. But Tremble is one which is submitted by authors, but it has not yet been verified. You will go for Tremble when you want to look at new sequences or something very rare sequences which have been very recently researched. Only those, if you want to have a look at those, you can go to Tremble. But it cannot be highly trusted because it hasn't been verified yet. But if it is mentioned Swiss prot, it means yeah, it has been completely verified and it is a good sequence and you can use it without any issues. Okay. Next is organism. It is coming from Homo sapiens, and here you have something called annotation score. Annotation actually in bioinformatics mean that you will be mapping the function of every gene which is present to a particular function. Okay. So, for example, so when we see a 5 out of 5 annotation score here, it means that the complete function of the insulin gene has been known, or the insulin protein is known to us. If this number is very less, say the annotation score is 1 out of 5 or 2 out of 5, it means there's only very less information available. So there's not much known about it. The higher score means it has been mapped well, and you have a quite good idea about what the function of this particular protein is. Okay. Next, protein existence. It shows evidence at protein level. It means that this particular protein has been identified in its protein form itself. So, in bioinformatics, one of the major issues is that not everything you see is an experimentally arrived data. Many of these actually includes computational data as well, which has just been predicted by some software, but it has not been verified manually or experimentally. Okay, so in such cases, you don't exactly know if it exists or not. That is why this protein existence is a very essential thing to look at to know how much of the protein is actually known, if it does really exist or not. Okay, so when it says evidence at protein level, it means the this protein has been identified in its protein form itself. If they say in the DNA level, that means only the nucleotide sequence form is known, but the protein form is not yet known, it has just been predicted. Okay, so that is how you will have several levels like DNA or mRNA level, and here we have the protein level. Okay, since human insulin genome is protein is like well known, we have a good score for this particular thing. Okay, now we have again so many other options in this. I'll just be briefing you on what all of these mean. Before that, what we would essentially need is the structure. We would like to know about what the structure and the sequences. Okay. So here, as I already mentioned, your three-dimensional structure will only be viewed in PDB. Here, you will be able to visualize it here. Okay. So now PDB is the one which will be able to show you the structures. So from UniPROT itself, you'll be able to access which PDB actually corresponds to your insulin gene. Okay, so that is what is showing you so many PDB IDs over here. The rest of them are called isoforms. Isoforms mean they are very similar to your insulin gene and they may have a very similar structure, but their functions might vary a bit. Okay, so that is called an isoform. Now, are the what usually in UniPROT, what we would mostly expect is the sequence. Okay, so this is the sequence of your insulin protein. So this is the amino acid sequence. As it was already mentioned, the length is 110 amino acids long. So you have 110 amino acids over here. So now when you click download, so this is the format you will get. As I mentioned, this is called the FASTA format because it starts with this symbol and you will have the name of this particular gene followed by the sequence. Okay. So that when you click download, this is what you will get. What we will usually do is we will copy paste this sequence into a Word document or a notepad and then we will work further with it. Okay. Now I am not going to close. Okay, I will come back to the sequence later. This just note down that this faster sequence will be utilizing it somewhere else also today. But just to give you guys, just to let you guys know what it is, I'm showing the rest of it here. And yeah, this is the mass. It shows the weight of this particular protein, which is approximately 11 kilodaltons. And I'll show you one more very interesting thing in the software. So if you go here, you will be able to see something called as a variant viewer. There are so many other interesting things to look at, but I will just introduce you to one. You guys can just search through and have a look by yourself. It's pretty much self-explanatory, but I would still like to touch upon some things. Okay. So a variant viewer is actually one which shows that in these 110 amino acids, if there is a change in any amino acid from one base to another, what would be the likely consequence? Okay, so that is what is predicted over here. So, for example, in your first amino acid, which instead of this methyl group, instead of this one, you have this amino acid substituted by G. Okay, if M is substituted by G, then what might happen is here, so this color here means an uncertain significance. But if it's substituted by V, then it might mean it is pathogenic. Okay, so for example, if you can click here, you can sorry, you can see what occurs if this M is changed into V. It could cause type 1 diabetes mellitus. Okay, and this has been verified by ClinVar, and ClinVar will be available in NCBI. You'll be able to see here. So this is the ClinVar database. So this has verified that if a change occurs in this particular amino acid to valine, then you will have type 1 diabetes mellitus. Okay. Like that, you have many such variants which are which have been identified, and their color codes have been given here. And if you click on each of them, you will know what actually those variations can cause. So for our and you can here it's also mentioned what the function of this particular protein is, and if you scroll through it, you will have lot more information regarding this particular protein. But for our purposes, what we will be mostly requiring is the faster format. Okay, so here you can get the faster, you can directly download it. But if you don't want, I mean, if you just want it for copy pasting purposes, you can just go to the sequence and isoform, and here click the download, and it will show you the fastest sequence. So essentially, this is what UniPROT is about. You will be able to get your protein sequence information. So am I clear so far, or do you guys have any other doubts? If no other doubts, please give me a thumbs up, guys. All right, that's great. Okay, cool. Now we'll be moving to our next bioinformatic tool, and that is PDB. So this is the most important tool which we'll be utilizing when it comes to if we want to do some experiments with the protein. Say for example docking. We want to see if a particular ligand can actually go and bind to a protein. If you want to see that, we would need the structure of the protein to analyze it. And PDB is the bioinformatic tool where you will be getting the structural information of proteins. Okay. So for today, I'll be giving you a very basic example. Let's take hemoglobin. Okay. So yeah, we'll be taking hemoglobin, and like I mentioned, here again you will be seeing what the function of it is and which organism it is in, and all of that. See, here you have organisms from gastrolophilus, intestinalis, but this is not exactly what we want, right? We want it in Homo sapiens specifically. So here itself, we'll be able to type out Homo sapiens hemoglobin. Okay. So here, all your keywords really matter. Uh, just a second. Hemoglobin. Okay. So if you look at this over here, you will be getting a variety of structures which will be mentioned over here. But and here you can again go for filtered search so that you'll get only your uh, Homo sapiens hemoglobin. Okay. But the thing is, all of these entries, each of them have a different format. One will be of one particular chain. You won't have the entire chain of it. So usually that will be an issue. But the thing is, it might be a bit of a difficult task for you to find the actual protein in PDB because there are so many other forms which should be utilized over here. The thing is, and for your docking studies, you will actually go for the crystal structure only. Okay, We'll be going for crystal structure, but here, if you can see, it is not the entire form of hemoglobin. We have an alpha chain, delta chain, and all of that. But oxygen transport occurs only in a beta chain. So again, you would have to go for a more specific search. Okay, so if you go for yeah, so oxygen binding hemoglobin. Okay, uh, just a second, guys. I'll be back. Okay, just a second. Can you guys hear me now? All right. Okay, let's carry on. So yeah, as I mentioned, you will be searching for hemoglobin oxygen transport. Okay. It is very difficult to get the proteins which we require because it takes a lot of time to get through what you actually want, because you have so many other substructures, and based on what specifically we are looking for, it will take a lot of time. So we'll just take the beta subunit. Okay. All right. Yes, so for example, we'll be taking this structure, okay, hemoglobin, and you'll have two chains over here. Yeah, as we go through, I will show you what each of this actually means, okay. So now this again, it is called as the accession number, okay. Like in every other software, we just saw what an accession number is. This is also the accession number. Yes. And here you will be having a classification on what its function is. So here, as we all know, hemoglobin is responsible for oxygen transport, and here it is present in Homo sapiens. So that is why the organism is mentioned as Homo sapiens. And the expression system, what expression system actually means is that when we need to get the experimental data for this particular protein, they would have to undergo a lot of experiments. They would have to express the protein in particular organism, and then from that carry on their research work, right? So here for this particular protein, they have used Escherichia coli, E. coli as the expression system. So they'll be producing this protein in E. coli and then using it for further research. Okay, it is important to know on which expression system you use it, so that uh, usually your expression system also has a lot of effect on your protein formation, right? Because you know how uh, you have post translation modifications required for a particular protein to function to its fullest. In those cases, they would be modifying the expression system accordingly in order to get the required protein. Okay, so that is why it is important to mention what the expression system was. Now, here if you see the experimental data, they have shown what experiment they actually used to depict the information which is being provided here. Okay, so here it shows X-ray diffraction. Most commonly used is X-ray diffraction in order to study proteins. Okay, NMR, X-ray diffraction. These are all studies which will help you to classify the three-dimensional structure of protein. Okay, so since they have actually performed the experiment, we know that this is a actual model and it is not a computational based model. Okay, that as I mentioned before, the one problem with bioinformatics is many of these structures could also be predicted by computational tools. So you may not actually know how accurate they are to the real ones. So if you have experimental data for something, it is a very good thing. Okay. So resolution, R value, R free, R value work, all of these are some terms just to show how accurate these are. Okay. So for example, your R value work should be less than zero point 0.2, or R value free should be less than zero point 0.3. If such is the case, then it means that it is very good. The lower your R value, it means the better is the result. It is more accurate. Okay, but you don't have to know about all of this. Actually, even I am not very familiar with all of this. I just know what I mean how these values should look like. Okay, So just know that there is a threshold values for these, and they should be low enough for it to be a good model. So when you want to know about the entire structure of this protein, you would have to go. These are usually the important informations over here. For I would actually like to give you one example. I recently worked on a project where we'll be working on human serum albumin. Okay, I hope you guys know what human serum albumin (HSA) is. If for those who don't know, it is a particular protein in our blood which is responsible for binding to various ligands. Okay, it has a high binding property, so and it is a very abundant protein also. Okay, so now in this protein, I had to do some docking studies. But if I am doing docking, I need to know how many chains are there against which I'm docking, etc. All of this I need to know. Okay. Here it is shown that it is a monomer. What monomers mean is that it is the exact same chain. There is no difference in those chains, and there is only one chain. One chain is actually called a monomer, right? But if you look down, you will have two chains A and B. It was mentioned it is a monomer, but you have two chains A and B. But if you actually go into it and see in the PDB format, it will be the exact same. So they are just two chains which are attached together, but they are the exact same chains. There is absolutely no difference between the two. So in such a case, what I would do is I would only take one chain, either A or B chain, and then I would perform my docking analysis so that I will get a particular score. It will essentially be the same thing. If I do for both chains or if I do for one chain, it will still be the same. But when it is mentioned it is a monomer here, it means that we have to take only one chain and do for you to have more accurate results. Okay, and docking actually takes up a lot of time. It's a very extensive process, and it will take a minimum of 3 hours to run just like 100 times docking. 100 times docking means you will be binding the ligand to the protein 100 different times to see in which particular position or time the binding is very strong. We are usually at small research levels we go for 100, but if you want to have much more accurate scores, you have to go for large number of runs. In such a case, if we take very big chain lengths, then it will take a very long time for you to get the results. So when we don't want to avoid all of those, we need to check for what the actual stoichiometry is and chain number of chains which are present, so that we use it accordingly. Okay. Yeah, I can show you further example in this itself. Here again, they have mentioned that it is through X-ray diffraction experimental method only, and here this will be the structure of the protein. Okay, and like I mentioned, for docking studies you would be requiring the crystal structure. So that is what you have, crystal structure of human serum albumin. Okay, and if you go down, you will have the entire sequence information about this. What is the name of the molecule? How many chains does it have? And what is the sequence length? Again, if you see, it is mentioned 585, so you may think it is 585 in both the chains together, but no, it is 585 in chain A and 585 in chain B, because it is a monomer and it is the same chain. Okay, and organism, it is present in Homo sapiens, and there are zero mutations which are present, and here you have the image. Now, yeah, this is also like a very important thing you could take note of, which is giving the structural information, like what is the weight of this, what is the atom count, all of this. If you all of these information you can get here. Okay, and okay, this shows sequence clusters. What this actually means that you won't be utilizing any of this, but this is sort of how your blast or any alignment tools work. So it just shows your 30%, 50% means what other proteins or sequences have 100% identity to this particular protein, which is human serum albumin. Okay, so that is what it essentially is going to do. And so if you have 95%, 90, 30%, so when you want to work with other molecules which are similar to this, usually you go for all the sequence clusters. So when this will come in handy is that some of the proteins have been only predicted using bioinformatic tools, and you don't exactly know what its function is, or how its structure might be. Okay, so it will be very difficult for you to predict all of this. In such a case, when you go for this identity, if you have, say, a very high, that is above 98, 99% identity with a particular sequence, you can assume that they may share the same function, or they may take up the same structure. So that is why your sequence identity matters a lot. So some sequences, when you have absolutely no idea, even the smallest amount of identity will help you figure out what could be the possible functions of at least some portions of that protein. Okay, so this is what your sequence clusters mean. And here again, you have, these are all some databases which appear. So we'll be looking at these in the upcoming sessions. Uh, Not all of them though, just a few of them. So it is just to give an information about this protein in various databases. So that is what this is giving you information about. Okay. So now, yeah, again, as I mentioned, this is giving you a detailed explanation of how the X-ray diffraction went. So you have all of this, and you have who submitted this particular version and how it has been verified. Okay. So, uh, yeah, Mahit, you can go ahead.
Yeah, could you explain sequence clusters again and their function? I couldn't get that concept properly.
Uh, yes, I will tell you. So, sequence clusters actually mean all those sequences which have uh, similarity to our particular protein. Okay. So, if you say you have 100% identity, it means the other molecule, which I mean, there are few other molecules which have almost 100% identity to our particular protein. So usually where this will be utilized is that when we have an unknown protein, which say we have predicted a particular protein from a bioinformatics tool, it may or may not exist in reality. Okay, but it is only being computationally derived. So you want to see if this actually has a function, if this could actually exist. In such a case, what you would do is you would try to align that sequence with another sequence to see how close they are related. If it shows 100% identity, you can tell that okay, since these two are very similar to each other, they may share the same function, or they may take up a similar structure. Okay, so that is what your sequence clusters do. Your percentage identity shows how close they are to your to another sequence. Your sequence is close to another sequence. Okay, so that is what a sequence clusters do. In the case of known proteins like say HSA, you will have many other proteins that share some similar functions or similar structures. But when you have very new compounds which are just reposited, in such a case, then if you are able to find any of these sequence clusters, you can try to predict what its function would be, and you can try to conduct a bit more research on that particular area. Is that understood?
Yeah, so just correct me if I'm wrong.
Uh,
So are they testing tools, or is there a particular list present when you click on it? List
no,
in the
there is a
sense
list
like
present.
what are the similar sequences, or is it a testing tool where you can test and store?
No. You will actually get a list of all of this. So, see, here, if you go into each of these, you will be able to see how many are actually there within this. So, in each of the organisms, like HSA like, then what will be that? So, if you click on each of this, you will get one other particular information. Okay. So, when you go into it deeply, you will have a lot more. It just shows you how many other members are that in similar to this, but we have others tools which will help us to actually determine what these sequences are. Okay, so this only gives you an idea about how much sequence similarity it has. And just a second, it will take some time to load because it is connecting with a variety of other tools as well. See, for example, here you can see all of these sequences have 100% similarity. Okay, so when you
Yeah, got it.
yeah, when you click it, you will be able to get what are there. So the thing is, it is also connected to another tool. Like I said, in the upcoming sessions, you will be able to see how we'll go for alignment tools to see how close two sequences are. You have something called coverage percentage, which means how much percent of your reference sequence is being covered by your target sequence, etc. Okay, so all of these will be covered in the upcoming session. But just to know what your uh, sequence clusters meet, the identity percentage just to show the other sequence which are similar to it. Okay. All right. So moving forward, yeah. Okay. So essentially, this is what your protein data bank is about. You will have the three-dimensional structure of the protein. So now, as I said, we'll be utilizing this for further other experiments, right? Say for docking or many other places where you will be requiring a PDB format. I'm sure many of you would have heard about PDB because it's one of the most commonly used formats. Okay. Before I move on to that, I would just like to show you the structure. If you go into the structure, you will be able to get the 3D view of this particular protein, and you will be able to visualize it with other softwares like PyMol, where you'll be able to visualize your proteins well. Okay. So in this, you will be able to see this is the structure of the entire protein. If you if you just run your cursor over this, you will see what are all the water molecules. Can you see the water molecules over here? Yeah, so these small round orange ones are the water molecules. So when you hover over it, those will get highlighted, and the rest will be the polymer. Okay. So this is when you have like the general structure of your protein. But when you have another compound like hemoglobin, I'll show you hemoglobin. We see hemoglobin is a protoporphyrin ring, as you guys may be aware of. So it has a heme group, which is an ion group in the center. So in such a case, it will have the ability to also bind to a lot of ligands, and it has a lot of water molecules also to it. So again, if you go into the structure here, you will be able to see a lot more water molecules compared to that, and you will have something called ligand. So these are ligands which are bound to the actual polymer. This is the main polymer, and these are the water molecules. Okay. So this will be the actual structure of the protein. You'll be able to download this and visualize it in other softwares like PyMol. There are various others to it, which we will go through in later sessions. Okay. Yeah. So now, as I was mentioning before, you need something called as the PDB format, right? So what you need to go do is you have to go into this download files. So the thing is, yeah, as mentioned before, your FASTA sequence will just give you your protein sequence. Now there's something new which has come up called as the PDB/MMCIF format. Okay, so what this actually means is that the PDB format which was there before had only limited information, but the CIF format will now have more complex information also stored in its also stored together. Okay, but we will only be requiring a PDB format in order to do this. So if I click the PDB CIF format, it will be downloaded as a .cif2, .cif file. Okay, but I don't want a .cif file. I need something called a .pdb over here. So in such a case, what I need to do is I would have to open some other tools like you can go for PyMod, or you have something called as OpenBabel. You have so many other tools in which you will be able to upload this and convert it to PDB format. Okay, so that is bit of a task. But if you want only a PDB format downloaded as such, and if you don't want any of your updated complex structures, you just want the normal structure of the protein, then you can go for the legacy PDB format. That will just get downloaded as your .pdb only. Okay, so this is the earlier version, and this has been used for like a very long time. If you want like a simple basic version, you go for the legacy PDB. If you want it with all the complex things associated with it, then you have to download the PDB MMCIF format, and then you will have to convert it to PDB using some other tools. Okay, so I can show you PyMol right now, but it will take a lot of time, so I don't want to waste time on that right now. So in upcoming classes, I can show you how the conversion actually takes place. It is a simple conversion, like how you convert your Word document to a PDF format. It is as simple as that only. There is nothing greater associated with it. So like that, you will just convert your .cif file to .pdb, and then you'll be able to use it further. Okay, so PDB is the repository from which you will get your protein structure, and you will perform your further analysis or further research. Okay, so I hope PDB is clear to all of you. If you guys have any doubts, you may ask. If it is clear, please give me a thumbs up. Alright, guys. So we are almost nearing the end of the session, and I will just be concluding it with one final software. This is like a very small one, wouldn't take up much time. Okay. So this is called as SOPMA. Okay. So you know how a protein has a secondary structure, tertiary structure, and all of that. Secondary structure, it involves alpha helix, beta sheet, coils, etc. Okay. So when you have a particular protein sequence and you want to know what its predicted structure could be, you don't know about this protein, but you want to know how it might fold in a secondary structure format, or how it might fold into a tertiary structure. So if you want to know all of that, you'll be using these tools. SOPMA is specifically only for the secondary structure prediction of a protein. Okay. So we need to have paste a protein sequence format over here. So that is why I told you we'll use this um, insulin sequence itself. So I'm just going to copy the sequence, and you can paste it here. Okay. So once you paste it, the number of confirmation states, we usually take it as 4. These are all called as default parameters. So we'll just take it as 4. And output width is actually to show how many, okay, when I show the output, you'll actually be able to understand that. Okay, so these are all usually default parameters. Okay, this will take some time, so it will say queue running, but you will be able to get the results in about 10 to 15 seconds maximum. It doesn't take that long, as long as your sequence is short. But for longer sequences, it does take a lot of time. Okay. And also, these are only like prediction tools to see how your protein could fold. Okay, so these are mainly used in your protein folding studies. So while we wait for this, yeah, okay, so we have got the results. So like I said, you remember I told you there was something called as the output width, right? The output width there was set to 70. It means in one line how many amino acids we are comparing. If at all it was like a larger protein sequence, it has many more bases, not just 110. It has a very big sequence length. In such a case, you would want to analyze only small bits of it. Then you can keep the default number to 70, so that in one line only 70 amino acids will be displayed. Okay, so that is what your output width means. So now your SOPMA has actually predicted what is the percentage of alpha helix, what is the percentage of a beta bridge, beta turn, extended strand. All of these are the secondary structures of proteins. And it is, yeah, so here it has calculate what the percentage is. Okay. So as you can see here, the random coil seems to be the highest percentage. Okay. So there are about 58 random coils in the insulin protein. Okay. Now it also gives you on which basis, which amino acids, what confirmation could take place. So which means under M, it is given as a H. So which means this could possibly be a part of a alpha helix. So this entire sequence could make up a alpha helix, and this entire part of the sequence could make an extended strand. This is E. What you will mention as E would make an extended strand. So like that, it is showing what possible structure a particular part of the sequence could take uh, by representing it with all of these alphabets. Okay. So this is what it is showing. This is a very basic tool. There is nothing much, nothing more information which you can get from this. It is only to predict what will be your percentage technical structures. And here, these are, as I mentioned before, these are only your default parameters. Your window width is actually to show how many amino acids it will take at a time in order to do the comparison. Okay, if you take a smaller number, then it will have bit more higher accuracy, but it will take a longer time. And similarity threshold is to say here it is taken as 8. 8 means it is to just show like how many amino acids at a time you would like to take to compare it with other sequences which are similar to it. Okay, so if you take like a larger number, then you can compute like the entire sequence. You'll be seeing how similar it is to another sequence, so that based on that, if its structure is already known, it will be able to predict the values much more accurately. If you can understand what I mean, okay? And number of states is four because you remember in the previous page we chose four: helix, beta, coil, all of that, right? So we chose four states, so that is why it's showing us number of states is four. Okay, so these are default parameters. You don't have to usually change anything in this unless we are going for very specific searches. So unless we do that, these won't change. So this is essentially what your SOPMA is all about. Okay, so this SOPMA is only for your secondary structure prediction. So, yeah, just to summarize what happened in today's session, I'll just show you all the software once, I mean, all the web tools once again. So, first one was NCBI, and in NCBI, you have like a variety of tools which are there, and these are the most commonly used ones, out of which our aim would be to use PubMed Central, Blast, Nucleotide, etc. Okay, so this is like a curated web tool with many information, and you have lot more to it. Actually, you guys can go back and look into each of these tools. They are very interesting to look at, especially the CleanBar database. All of these are very interesting to look at, so if you are interested, you can have a look at these. If at all required in your upcoming classes, we'll try to utilize any of these databases and also work. Okay, so first one was NCBI. Next one was UniPROT. UniPROT is only for protein sequence analysis. So if you want to get information about your protein sequence, like what amino acids make up this particular protein, then you will go for UniPROT. And from UniPROT, you will be able to download the FASTA format. Okay, FASTA format is the one which will show you the sequence of amino acids. So this is the one which you will be copy pasting into other softwares if you want to work with this particular protein. Okay, so that is what your UniPROT does. And just remember, I hope you remember all the parts which I explained over here, what your Swiss prot is, what tremble is, so what each of these mean, what protein existence means, what annotation score means. So if you have any doubts in this, you may ask me. And there are so many other aspects to it as well. You can take a look into those. They're very interesting. But since we don't have time today, we'll be stopping it till here. But in upcoming classes, if we'll be utilizing any of those, I'll be explaining to you what those mean as well. Okay. So that is about UniPROT. Then we have PDB, which is Protein Data Bank, and this is to get information about the three-dimensional structure of proteins. Okay, so from this only you will be downloading your .pdb format, and this will have the structure of the protein with which you will be performing other bioinformatic approaches on it. Okay, so this is what PDB does, and it is based on experimental data like X-ray diffraction or NMR, all such of it, and you will be able to visualize the 3D structure as well once you go into the structure. Okay, so you have a lot more information about this available here in PDB. So PDB is for three-dimensional structure of proteins. And finally, we covered SOPMA, which is for the prediction of the secondary structure of a protein. Okay, so it will align it with existing proteins if it has a similar structure. Based on that, it will predict the particular structure to see how what percentage of alpha helix is there, what is the percentage of beta sheets, random coils, extended strand, etc. Okay, so that is what SOPMA does. So that is prediction of secondary structure. Okay, guys. So. Do you guys have any other doubts? If you have any doubts, you may ask me. All right. Did you guys find this session useful? Were you guys able to learn anything? If so, you can give me a thumbs up. All right. So I hope today's session was easier to understand. I'll just stop it.